Okay. So just on the sort of the housekeeping, so we've had a change in our agenda. We were going to have Jaime. He couldn't make it, and so I'm trying to grab him for next time. We also have Judith for next time, I hope, on Marichelle, I think is her, how you maybe say it, who's doing a bunch of sanitation work way up in Hudson Bay. So she had presented before, and now she'll have some more to talk about with how things are working up there. So that's for next time. And Jasmine, you're here, and I think, did we talk about you next time as well?
Yeah, I'm
Hey,
open.
all
I'm
right.
open to share all the news that I have. So
Great. So I'll be in touch about that. But meanwhile, our agenda is basically, so that's we had some great discussion last time. Just looking at my notes, it was the fellow from Alaska talking about anaerobic digestion. Well, first of all, good update on the test anaerobic digester NREL EPA project in Fairbanks with Annie. Maybe she can talk about that when her time comes here. And Wilder, you're involved on that. So I guess between the two of you, maybe we can get a super quick update via introductions on that. We also had the two people from Alaska, Archilla, Sean, and the biomass guy at the Alaska Energy Authority, and the state energy coordinator for USDA Rural Development, Dan Smith. So that was great. And those were our two big things. So I guess, and we talked about this fish waste processing thing in Alaska. So that was interesting. Let's do, how about we move to introductions and then we have our very brief agenda possibly. So introductions and updates, please. Shall I do it in reverse alphabetical order of last name, or how do you want to do it? How about that? We'll do, looks like maybe we'll do Brown followed by Corrigan, followed by, no, Brown followed by Bronsted, followed by Corrigan, followed by, we'll just, and then, sorry, Brown, Bronsted, Bywater, Corrigan. Can we go like that, please? Introductions, updates.
Great. Yeah. I'm Wilder. I'm the intern up at NREL in Alaska, working with Annie on the anaerobic digester pilot experiment. Coming up here from Vermont for the summer. And let's see, I don't know how much detail I should go into for the project right now. How much time?
Wilder, I will say my last update that I provided was that we didn't launch. So I think everyone is up to speed on that unless you weren't at last meeting. So a quick 411 is that we didn't have the budget to do the full operating season. So we are not full of sludge right now on site. But we have the drums full of water and the power system is on. So we're collecting temperature data across the 4 different systems at least. And so Wilder, you can maybe start there to talk about that.
Yeah. So we have the 4 different drums filled with water right now. One of them in like a passive solar greenhouse, one of them with the like thermal heated blanket, one of them with kind of an ambient like resistive heater inside a shed, and then one of them outside as a control. And let's see, should I pull up just my plot of the data maybe, or?
Yeah.
Yeah,
All
we've got
right,
time. I
sounds good.
think our agenda is just this whole check-in and then EPA's sort of publications database, and that's it. So, I mean, other than that, so I would love to hear things. Now, how do I give permission to share, or is that already possible?
I think it's already possible.
Great.
Okay, let's see here. Oh, is it working? Can you guys see this?
Yeah, we're
Here
getting
we go.
there.
There we go. I
Nice.
think. Okay, cool. Yeah, so this is just a couple days worth of data We just recently started collecting like a week and a half ago. And so there's been some hiccups trying to get everything working. But we're seeing data, we're seeing kind of the performance that we'd expect. The green line is the heated blanket, so that one is by far like the most consistent temperature wise, but then we're seeing like the control versus actually at the point that this data was taken, the orange line, the drum inside the shed with like a resistive heater, was not turned on yet. So we're not really sure about the impact of that yet, but yeah, starting to collect data, and that'll be part of my work over the summer is just getting the temperature data, because that's all the sensors that we have set up right now, but trying to parse from that how well the bacteria would be able to survive and produce biogas potentially. And yeah, I think that's about it.
Do you have
So-
energy data for each of these? Like kilowatt inputted?
What was that? Energy data?
Yeah.
Yeah, so that is gonna be part of it too. I think we have, so the two that are connected to the islanded solar battery bank are the drum blanket and the resistor. Currently, like pulling data, we have like a very old solar controller thing. I forget what it's called, but I think I'm still working out how to get the data off of it, but it is collecting data. So I'll be able to, yeah, see kind of the amount getting stored to the batteries and the load.
Yeah, that'll be good to know. It looks to me like, can I ask a question, I guess? Anybody else? I'll yield. People can raise their hands or just ask.
I just yelled.
So it looks to me like the orange and the blue are basically what the drums are doing. Neither of them is energized. So the sort of the red, actually the red line is what's in the just greenhouse ambient air. But the orange and the blue, no, orange is really the representative drum temperature.
So the orange is in that one. It's not being heated at all yet in this graph, but it is inside of like an enclosed shed. So there's kind of less airflow, and it's not getting direct sunlight. But the blue one is fluctuating a little bit more because it's outside and getting direct sunlight for part of the day. So.
Oh, that's a drum, that blue thing.
Yes, yeah,
I
sorry.
thought that was outside ambient.
Oh no, so that's a drum. That's like a control drum, but it's outside. Yeah.
Cool.
Cool. All right, well, I can stop sharing if anyone else has questions. Let me know. But yeah,
And
nice to. Oh,
I was just gonna say that there's also, so Wilder also is collecting the air temps. So these are all the drum temps, like water in the drum. And so there'll also be plots for comparing what the water in the drum is to the air temp. Just to have that comparison too. These will be able to be built out. We're also doing some finagling because the sensors are not exactly on their intervals for collection as they should be. So Wilder's been awesome at troubleshooting, making sure it's working as we expected it to work. Thank you
So
for
when
presenting.
I look at that line again, I'm looking at the thing that's labeled greenhouse. Annie, you said all the temperatures are in drums. What does that red line indicate? Greenhouse.
Oh, that's in the drum also. These are all in the drums.
Ah,
Yeah, so none of the ambient temperatures are plotted yet on this one. Cool. Well, thank you guys. I forget who is next.
I saw your message, Nate. You can maybe jump in
Yeah,
line.
just for ease of time, maybe I'd like
Yes,
to
yes.
jump in. And also this is a great pivot so that you're not just sitting on your hands. It's great to get some data, and that might actually help you tweak the system in little ways that could help once we actually seed it. Anyway, that's great. So my name is Nate Janiga. I'm a process mechanical wastewater engineer focused in water reclamation and reuse. And I've previously worked for, 6 years doing membrane bioreactors. I am trying to get out of that brainwash tunnel vision to be more of a generalist. And I'm learning a lot of stuff while working as a consultant with Carollo engineers in Seattle. I really like that. And I've gotten to work with, these fine people and learn a lot more about what's going on in Alaska and what's going on with, anyhow, everybody knows that part. This is a great team. I'm really happy to be here, and I look forward to finding a way that we can run our pilot study next summer. And I'm gonna come back after about an hour or less, maybe.
thanks. I think next would be Embre, and then Angie. Yell.
Oh, I hate this part, Alex.
Okay, skip it. Put something in the chat.
Okay, I'll do that.
Whatever.
I'll chat. I'm the opposite of Nate. I am a total generalist, complete and utter total generalist. I worked as a consultant, and then I worked for a general contractor in the field, and then I worked for a university. And so I know a little bit about a lot of things. And so I work for Nate's competitor, Brown and Caldwell, also a consultant, where I'm mostly a PM right now. So fair?
We collaborate sometimes too.
Yeah, like on West Point. Yeah, we're supposed to be collaborating on West Point. Should I review your SOP, Nate? For your biogas?
I'm sure that's gonna be out of scope for King County.
Okay, a lot of action there in Seattle. Over to you, Angie.
Hi, I'm Angie Bywater. I've had my own micro AD, I suppose, for about 15 years now, and we had a company that used to build them. We shut it down a couple of years ago. I represent a trade body in the UK as an academic member, and I ran with a foot in academia and a foot in industry. I was running the Anaerobic Digestion Network, which was a funded project for that was essentially 11 years in various guises. I'm now working on something called the Global Center for Sustainable Bioproducts, but I do keep a foot in the anaerobic digestion side. So I just there was a couple of things that people mentioned, and I wanted to bring it to your attention. The IEA has just published a manure report, and it's kind of slightly off of what you guys are interested in here, but that's IEA Task 37. I can drop the link in the chat if you like. And it occurs to me when Wilder was talking about the monitoring, we have a paper that we did like in London 10 years ago when we were running this small AD, and it does talk, the AD is inside a greenhouse, and so many of these sort of things, and we did run it, I think, a bit on solar, so a lot of these things might be helpful. So I'll drop a link for that in the the chat as well. And if you can't get hold of it, somebody just email me and tell me, and I'll just send you the PDF of it, because I don't know if it's behind the paywall.
That would be great. Thanks so much.
Yeah.
Okay.
And I guess, Angie, I didn't know your background on the microbiogas stuff. You know, as we proceed here, depending on your interest, maybe you can do a little segment on what your work was and sort of what you learned that would be applicable to this group. Yeah, so
Yeah.
we can talk about it offline, but I, you know, you just good information on microbiogas in a cold place is hard to come by. So I'm really curious if your experience will be something that you could share. And we can, of course, put your stuff in the repository, but it would be great to maybe have you present something as well.
Okay, yeah, not a problem. I did talk a little bit about it when we had our face-to-face meeting a couple of years ago. So, but yeah, if it's helpful to other people who weren't there or whatever, happy to do that.
Yeah, I was not there, and I think, yeah, the more we can get stuff here and online in our repository, the better. Yeah, super interested. I don't know if you were here for the Swiss people.
No.
So the Swiss people, I put their presentation in our Google Drive. So
I think I'll have a look.
yeah, that's around too.
Super, great.
Yep. See, sorry, I'm alphabetically challenged here.
Me, I think it's me.
Good. Yes. Oh, yeah. If you want to go again, yes, please.
Yeah, I think I don't need to take too long here because I know everybody, but I'm working at the Fairbanks pilot experiment. And Angie, it is good to see you. I was thinking of you actually last call because some of the conversation had pivoted to what to do with the digestate. And I know we talked about using it as a fertilizer. But when it came to as a business, if there was any sort of like on a small scale, I think there's no issue with permitting, but there was a question around if someone were selling it as fertilizer, what they would need to do if there were other, like proving what pathogenicity had declined or et cetera, or if it were sold.
Yeah, we always struggle with that sort of thing from a small scale, because in the UK you have to reach end of waste, and end of waste is sort of wave a magic. Well, this is Angie's kind of slightly biased interpretation of is wave a magic wand and pay a lot of money and you kind of get end of waste. And particularly where there are potentials for pathogens like with human manure, or things like that. I can put you. I will drop in the thing what they look for end of waste, because there's a lot of stuff in there that they keep readdressing and trying to tweak, mostly about pathogen levels, but also residual biogas and those sorts of things, just to make sure things are properly digested. So for us, it's small scale in most of Europe under the Renewable Energy Directive and all the other stuff that we're covered by. Yeah, you need to make end of waste, which was a shame because it meant for small scale AD, for the micro scale stuff, they didn't give us what I would call a low risk position to say, actually, in small quantities and used under these circumstances, it would be really good to do. They wouldn't do that because obviously there's no money in it, but that's just my slightly jaundiced opinion.
Sure. Does that mean that it ends up being kind of cost prohibitive to get to end of waste and allow for that to even happen?
Yeah, for the small scale guys, it really did, which was a shame because many projects in London that we had, and indeed in farms that were doing sort of nice circular stuff, it meant that they were really worried about using their digestate, because unless we had enough excess energy to pasteurize, then they were really hung out to dry, because if they sold some food that was contaminated because they used this digestate on it, they wouldn't have a leg to stand on. So they were obviously very risk averse for their businesses, and we couldn't get them to do an aggregated position. So let's say you had like 6 of these and you wanted to just randomly sample and just aggregate it, then they wouldn't, nobody would consider that, because there weren't enough small scale guys to do that. But then on the other hand, there couldn't be enough small scale guys to do that because they couldn't get over this end of waste. Personally, I've used unpasteurized food waste digestate and cow slurry digestate on my garden for 15 years. And in the same way that if you were making a herbal soup, you wouldn't throw it on your lettuce leaves, you wouldn't do that with digestate. So, you know, common sense needs to apply, really, in some cases. And the stuff that Jasmine was using with the dog do, there was even worse things where, because potentially there's potential pathogens which, when put under climate stress, when put under stress, will form oocysts, and these things will not break come hell or high water. So you know, there are certain things that you have to prove a relative degree of safety. In food waste, I don't know about human waste. Jasmine, you were nodding. I haven't looked at human waste in those respects, but it was mostly dog waste issues in those oocysts. So the other stuff isn't so much of a problem.
Goodbye to Nate. You'll be back. I love this discussion. I think, so I have a couple of things to add, but can we have this as an agenda item? Because obviously the point is to, you know, manage waste better, but then what do you do with this stuff? This is the blind spot on so many of these projects, because people think about energy. We're not in that space. We're thinking about sanitation, and this as a means to sanitation. So not as a means to energy necessarily, anaerobic digestion, that is. Before I forget, Angie, yes, please put that link on the IEA Task 37 in. But I would like to hit this topic again at maybe at our next meeting. And Judith may have some insight on this. I can say that for us in America, there's a federal rule, and you all know it better than I, but there's called a process to reduce pathogens, and that's a list of things that you can do, everything from liming to anaerobic digestion to composting. And then there's process to further reduce pathogens, which is another set of things in Code of Federal Regulations, Chapter 7, 503, whatever it is, right? So it's all about, if you do these methods, then you get to a PRP or PFRP. So that's kind of how it works. On the food safety side, the way it generally works, as I've observed it, is that it's like ISO 9001. Say what you're going to do and do what you're going to say. In other words, document what you are doing for food safety, what's your plan, and then show when somebody shows up that you follow your plan. So, you know, how do you wash your vegetables? How do you pack them? What do you do about refrigeration? What do you do about irrigation water? Ditto this. What are you doing about digestate on your land? You'd have to have a food safety plan that incorporated it. Forage, different story. But anyhow, that's my understanding of how this works in the United States. We also, as the Agency of Agriculture, regulate soil amendments, and biosolids is not in our camp. That is at the wastewater treatment people. So that's a whole different, that would go over there. If it's manure, we're gonna regulate how it's land applied. Or if it's a product being sold, it's got to be registered. And that's all about disclosure.
Thanks, Alex, for adding that. Closing the loop on kind of in response to Angie's comments, I was thinking about the sanitation as the means for the motivator, and a little bit looking at Mark Orell, because if we're talking about remote Alaskan deployment, that's definitely a question of, well, when you have the digestate, do you just put it in another lagoon when it's, you know, is it just another thing before still a lagoon? That's part of the question. If we aren't using it to grow food, if we're not allowed to have other uses, kind of what can be circular or just not? How much, yeah. If anyone has comments. Otherwise, Alex, we can totally talk more about this on agenda.
Yeah, there's
ahead.
definitely a lot of potential for, I think that's like a big driver, especially for this group in my own research, for sure. Why I got into anaerobic digestion, especially for rural communities in Alaska. The digestate that would be like coming out of an anaerobic system, especially for most human waste, it would be ideal for it to definitely go into a sewage lagoon, right? But then there's also a lot more room for, you know, community-led research in terms like if the community is interested in having anaerobic digestion and how it might apply to them in terms of maybe like applying it to their land, doing some research there. You know, here in the Midwest, I've been learning a lot for wastewater, especially. There are a lot of cases where we're doing anaerobic digestion and then we're doing putting the biosolids on farmland. I haven't looked too much into that extensively here, but I do know that it's happening. But yeah, I think that there's a lot of potential there for sure, but ideally probably in the sewage lagoon and it would be good to go. But of course you could like filter it through charcoal. You know, there's some things that you can definitely do to enhance it. In my opinion, it's better than a lot of the systems that people have right now, right? Because, you know, a lot of communities have to self-haul. So
And the tightrope we walk in Alaska, really, I think that a lot of states walk is the moisture content question. I mean, up here, we can't dispose of anything that comes out of any digester of any sort, really, unless it's in some kind of a sewage lagoon, specifically if it's of a certain moisture content. And our problem, of course, is to de-water the stuff, right? Includes either, you know, huge amounts of energy or giant amounts of polymer. And, you know, it's a massive challenge, especially in a small community, which also typically doesn't have, you know, enough agriculture to even consider land applications for any kind of use other than disposal. And the state, without a lot of testing, my understanding of state regs is they're just not going to accept it for anything other than deposition in a sewer lagoon system or final placement in a landfill, as long as it's classified as such, and those are difficult to get classified. A landfill for that kind of thing is difficult for a small community, cost-wise and management-wise. So I'm Mike Marcarell, by the way. And
Okay,
I love this group because I like to be in a room where everybody's smarter than me, and because that's how I can pick things up. I keep those article references coming because I wish I was more of a generalist, like Embry pointed out, but I'm more of a specialist. I work for the Alaska Native Tribal Health Consortium. I mostly do senior review of engineering planning documents and designs, some construction planning and some construction inspection, but mostly I'm focused on water and sewer. And I'm not strong on microbiology, but I am a fan of making solid waste turn into gases for multiple reasons, and especially in small communities and in cold climates, which has been a stumbling block for me for years. That's all I say, I think. Back to you, Alex.
Yeah, I think we're going to have to organize this little talk. This seems really important to me. I'm going to circle back to the people who are talking about it here to see what we should bring forward on it. Seems to me just a very basic, maybe like a very basic go round, and maybe Judith knows Canadian regs, you know Alaska regs. I have a general idea for manure, but we're not talking about manure. It doesn't help. I don't know, like a brief session on this would be, I think, really helpful. Just what are the regulations and what's practical. If you think about having gone through a digester and then sitting in a sewage lagoon for X amount of time at some non-frozen temperature, it seems to me you're going to have a very low pathogen load there. But what does that get you? I don't understand it. So what does that get you in terms of regulation? I don't know. If we can do that information session, maybe that's one. And then we could follow up with, let's try this, or what are the likely good scenarios coming out of that? Because it would be nice not just to talk about, these are the regulations, these are the challenges, but to talk about what should we try? What should we do? What is a promising way to proceed. Yeah, that's where I want it to go. All right.
Can I just weigh in and say, I
Yeah,
think
yeah.
we should think of something else to do besides put it in one here. I think that there's still value in that.
We should think, what? Say again?
We should think about other things to do with the digestate besides put it in a lagoon. It still has value.
Yeah, I mean, it sounds like that's the challenge. Like, what are you allowed to do? What's safe to do?
Char it and use it as a fuel source.
I don't like losing those nutrients, to tell you the truth. That's my, soil amendment, like healthy soils thing. And charring might be the way to go. I mean, there was that company in Connecticut that makes sanitation systems that are based on charring. I'll send you the link for that. So yeah, that can certainly be done.
You're talking about biochar?
Yes.
Or just dry, right? So we had a project where it wasn't digested sludge. It was relatively raw and that still had a lot of organic content in it. And we dried it and we're using it in kilns, right? Like every rural Turkish village that I've ever been to in my entire life, where they have goat poop patties stacked up outside their house and they're using it in there to fuel an oven, right? But there's there are companies that are taking poop and hydrothermally charring it, not biochar, but hydrothermal char. And then this is one of my favorite things ever. It has the same caloric content as like brown coal, right? That's not a joke. That's actual like BTU per kilogram of this hydrothermally charred poop has the same energy content as brown coal and is a potential fuel source. So I take your point, Alex. There's nutritive value, there's nitrogen and phosphorus, but if there's not a ostensibly obvious place to land apply in Alaska, that there's still other things we can do with it besides lagooning, potentially. That's all.
I'm going to drop the link to the Connecticut company that does this for sanitation. Biomass controls.
And if there's any feedstock from the solid waste side that could be mixed in with it, that's up, again, like there's the potential for commingling of feedstocks to generate a product with a certain caloric or nutritive value.
Well, how about this? Maybe what we could do, Margaret, you're up next, or Michael, rather. We haven't actually heard from you. How about we do this? Michael, just to, you sort of introduced yourself already. And then let's go to Robin and then Margaret, because then Margaret can segue to this EPA thing.
Not sure I have anything else to present. I'm mostly watching others move things and trying to find ways to support. I think my intro was all I need to say for this current period. Thanks.
So then Robin.
Okay, thanks, Alex. Yeah, I'm Robin. I'm based in southwest Scotland, next to the castle that you see in the picture behind, which is just our local scene. So to put that in perspective, yeah, I've been on this group from the beginning and always enjoyed it. But given the punishing schedule that I have from work from various times, it hasn't always been possible for me to join. So I have had a bit of a gap. principally, I've been my day job has been to work on large scale anaerobic digestion facilities. I am a biologist and an organic waste specialist with our own specialist company in design and concept systems, conceiving plans for large AD plants. That's as I say, the day job. The smaller stuff, community projects and so on, are just part and parcel of what we do. It's always been our philosophy while working on big stuff, always to try and keep on board with R&D and other things of a more community base. I was convener for the NATO meeting, the NATO sponsored meeting, which was referred to earlier, particularly by Angie. So that's something to come back to in future discussions. As a biologist, I've always specialized on both small and big projects, in particularly the biohazard side of things. So what we've just been talking about over the last few minutes in terms of regulatory impact of digestates has always very much been close to my heart. And with projects that I've done across Europe, particularly Scandinavia, projects in US, North America, and then Alaska being, if you like, off to the side of that, I'm very, very aware of these issues. Europe has been actually much more strict in terms of management of biohazards than the US and to some extent Canada, but much stricter in Europe than has been the case under, for instance, EPA Part 503 regs and so on. So I think life is actually easier in terms of getting material into use and really winning back that value from digestates. It's almost impossible on a small scale in Europe, as Andy mentioned, but that's something that I'm happy to contribute to in the future. The other side task that I have is that I'm an external lecturer and board member for the MSc course on renewable energy engineering at Aberdeen University here in the UK. So that's something that gives me an academic link, and that's the academic side is something that's always been kept close to my heart. So in terms of where we go from here, I'm actually stopping a lot of my purely commercial work because it's time to work towards retirement. But that doesn't mean that I don't want to continue to contribute in the public good projects, of which this is very much one. So that's all from me at the moment, but let's see.
I would say exactly the opposite. Now that you're retiring, we're going to latch on to you.
I know. So I have to keep a proportion of my time for these things. So there has to be grandchildren to look after. There has to be a little bit of kind of, what can you call it, debris from a whole career working in the AD sphere. But public sector, public interest, public good projects, I'm happy to keep a proportion of that. And if we can find a way forward from my aspect under the guise of the Aberdeen University connection, which I'm keeping, then we can do something. The issue will be money, and not just for me, but for everybody, how we continue to search for funding for this group, because talking is cheap, doing work and getting projects functioning and actually valuable and worthwhile, that's the problem that we still have. It doesn't matter where we are. Even if I come up with a series of good projects for students at the university, that's great, but we still need to find money for bench time and everything else. So that's the real challenge that we have. Okay, thank you.
And I know we're being recorded, but good luck finding funding in the United States right now.
I know.
Well, we had some sort of positive noises from the Alaska people about, could they help fund or would they know a place for funding for for the NREL pilot. So yes, you're right. This is what we always are thinking about. Yeah, and I think as time goes on, we get better and better positioned as this group to go out and know what we're looking for and who's going to have it. I feel like I spent a lot of time diving into the potential funding before I knew sort of what our challenges were. So now that we know that better, I think maybe it'll be, yeah, I think we can direct our efforts a little better.
Well, do you mind if I continue with another question? And that is that because I've had time away from this group, did we ever get a written answer from NATO for the next round application?
No. Bupkis. Both Jane and I have been sending emails to the contact email as from the application, and we've gotten nothing. No, not even a, we received your application, we're reviewing it, it sucked, nothing. We got nothing. So, I mean.
Okay, well, would you like to give me the job of actually trying to speak to somebody there on behalf of the group? I'm happy to do that because I'll have some time over the next 3 weeks or so, 3 or 4 weeks, when I can maybe do that. But I don't want to tread on toes. I'll take that on board if you wish me to do so.
Afraid of treading on your toes by asking you to do that, Robin. So we've been avoiding asking you, honestly.
Okay, I will do it.
Saw a sneaker.
Okay, I'll stop talking before somebody gives me another job.
You just gave yourself that one.
Yeah,
You're already back and you're at
No,
it.
I know, but I'll stop now. Okay, I'll do it. I'll do it.
that's great. All right, Margaret.
So I am honestly heartbroken about the Alaska NRL digester situation, but this meeting and all of you are making me feel a lot better about stuff. So thank you all. And in the interim, I'm hoping it's an interim, where we don't have poo in the digesters. The thing I can think of to do is to work more on what has been brought up a couple of times, which is the regulatory context for some of this work. And I'm interested to know that maybe Europe could also use some, I'm not sure, work groups, documentation of how to make it work for the small scale as well as the larger scale. One of the issues that sort of brought this whole thing to my attention originally was that, as I understand it, EPA can only fund proven technology, and the ways of proving things tend to be done in more temperate climes, what we call the lower 48. And so I realized that I should probably start getting our work into some of the places where it registers as acceptable. And one of those is the EPA Searchable Clearinghouse of Wastewater Technology. And so I'm gonna stick a link to that in the chat. And I decided to start with offering through the public access page the journal article that we have from the very first phase, the bench scale test, and say, hey, this is an EPA thing. This is an EPA database. Why, maybe we could have it. And just as I had hoped, the folks at EPA headquarters were enthusiastic and got back to me and said that they would very much like, they agree that there's only the anaerobic digestion information in that clearinghouse is only for larger scale and is already pretty limited even for that. And so they are enthusiastic, and some of them may or may not be on this call or off. We're trying to invite them about having more information on small scale anaerobic digestion. And so I'm gonna spend some time trying to figure out what sorts of things make sense to include in the clearinghouse. And I'll be, including Dr. Marti papers and whatnot if I can. But all of you are, if there's things that you'd like to stick in, it seems like we have an invitation and opening for that. So that's, one place. Mike might also have suggestions, but, he and EPA work together on, or traditionally, who knows what the future is, on sort of, what's approvable, what's fundable. And the person at EPA who was in that role for a long time has taken the, whatever it is, let's hope that the legality of it doesn't pull the money back from his bank account. So there's sort of a, I don't know, institutional gap there. But there's probably, I know may not be, anyway. So yeah, I feel like there's opportunities to try to help build supportive regulatory structures for this work. And so that's something people have suggestions on where they think it is lacking. That's something I think I can tackle. The current presidential administration is in favor of streamlining permitting, for example. So that might be something that works for everybody.
So I love this, and I love that you're going to put stuff up there. Do you want to show us it by sharing the screen, or would it be something where you might be the curator, and we could possibly throw things into the Google Drive, and then you could decide what to put in the clearinghouse? Or I'm sort of reluctant to go in there, A, understand it, B, put things in there that wouldn't be appropriate, and it's just a bunch of junk, right? So I don't know how curated this thing is, how you want to handle it. I would be totally comfortable dumping things into our Google Drive, and then you decide.
It's pretty curated. You can't just dump stuff straight in. So that you can not worry too much about that. But whether you guys want to submit directly or whether go through me or the IARPC cold high anaerobic digestion Google Drive, those are all fine. There is some work involved, and I feel like it is work that makes sense for me to be doing, but I also don't need to be an extra barrier along the way. So in my sort of poking around, I think, there's a couple of categories of information, and on-site and decentralized is largely where we'd be. And so
I love that URL.
One thing whenever you end up with a bunch of dots in it, you have to check to make sure that your computer is reading those correctly, I have found. So it's a little slow. So there's two things currently that show up in the on-site and decentralized that contain anaerobic. So you can see the current information is somewhat limited.
No wonder they think that anaerobic digestion is not proven.
Yeah. So.
Well, what I'm gonna do, I think, Margaret, is let's put a little pin in this for the next meeting as an update, just a short agenda item. But I'm going to go into the Google Drive, try to organize a little bit more all those photos that you have. I'm not sure where they belong, but I'm going to throw some articles in there that you may not have. And then you can just go nuts as far as uploading stuff from there. That's my thought. And maybe I'll email you and say, hey, I put this thing in the Google Drive. I think it's really cool, but maybe it's not, or whatever. And just let you know that what I found of value. For example, I have a study that I paid for on the life of fecal indicator bacteria in a digester, and pretty interesting. Everything gets destroyed except Enterococcus. So, weird stuff, but interesting. Like, here is the actual lab result on pathogen destruction.
So yeah, in my mind, in this period when funding is so uncertain, pivot slightly to working on making sure that the regulatory structures are in place for when funding may appear, is my thought.
Are there any other ways that we can help support whatever you're doing right now? Or, yeah, obviously Alex is going to help support this effort to fill up the clearinghouse and show that AD is a real technology. But anything else?
What Mike was saying earlier about difficulty of finding ways of disposing of things. I've always sort of assumed that Alaska gets a waiver from everything, but apparently not. So, and it is true that there's been a number of bumps along the way where the National Renewable Energies Lab has suddenly said, we need a biohazard, removal plan. And I've been able to line up the Alaska Department of Environmental Conservation and us and be like, where are the regulators? And we can, verify this isn't a biohazard, you don't need a biohazard removal plan. So, identifying where people encounter these friction points and then try to work with the regulatory authorities to document that the friction points are unnecessary, I guess, would be something I feel like I can attempt to be working on. And if people identify where those friction points are, that's very helpful because that helps direct that attention and energy.
So I'll put this as a short agenda item for next time. And, run it past some people who have been in this conversation and could be in that conversation for what a short item would look like. What else? We're at 1 hour. Just checking our agenda here. Did we cover most stuff here?
I didn't go, but I'll just be really brief because I'm gonna go over it in the next meeting. But most of you guys know me. I'm Jasmine. I'm still with Zender.
We haven't had you yet.
Yeah.
Sorry.
No, you're okay. You're totally fine. I was patiently waiting. I wanted to hear from everybody else, but I'm Jasmine. I am affiliated with Zender still, but I am currently a master's student going to my PhD here at the University of Kansas, where I'm continuing some of my anaerobic digestion work from my undergraduate that I presented to some of you folks in Alaska. I got the fellowship through the National Science Foundation and then another funding opportunity through the University of Kansas. So basically what I'm interested in is collecting microbes or buying relatively the same microbes from a depository. It really just depends on resources and if I'll be able to go back to Alaska, but psychrophilic methanogens, and then seeing if they can, digest human waste at lower temperatures is really what I'm trying to aim for. Yeah, so I got that funding and I'll be doing that research for the next 4 years. I'm also currently for my master's working on this modeling of anaerobic digestion. It's called ADM1, Anaerobic Digestion Model 1. And has been really, really useful and interesting. I'm not too sure if anybody else has heard about it, but you're just modeling an anaerobic digester. And so I think over time, as I get more familiar with the model, I'll probably try to model a similar system that I have back in Alaska, but try to incorporate some of the data that I gather over the next couple of years. But yeah, some exciting stuff on my end. It's pretty intense right now in general, but I haven't been affected in terms of my funding. So, you know, I have high hopes for the research that I'm doing. My anaerobic digester in Alaska, I checked on it like last week. I was in Alaska for a few days. Some pretty great stuff still happening. The Alaska Pacific University, they're still using it. And in fact, they're using it as a teaching tool. So when I went out there, there was a lot of, there was like a little station for people to come in, burn the gas, and, you know, have a cup of coffee or tea, hot chocolate. They bring kids out to the farm and they let them just look at the digester, and then they all have like hot chocolate together. So I think that's really great. That's kind of what I was envisioning. Originally I had gotten some backlash from the university for that digester. So it's great that they're, you know, accepting it, kind of the things that we're hearing right here with regulations. And since I received the funding, we're going to be looking more into just developing that relationship a lot more, and just trying to get the university more involved in getting more youth out there to check it out, especially the ones that they have like camps out at the farm as well. So yeah, I'm excited. I just started the journey, but I'm excited, and I'm really happy to still be a part of this group. And even though the EPA, the digester in Alaska, you know, there's been some roadblocks, I think it's great that we're getting that temperature data, because I also have some temperature data as well. And so I think it's great that you guys are getting that. But yeah, I'm excited. That's
Two
it.
questions.
Yeah.
One is, maybe so first of all, what the heck is this new model? Is there a link? I have not heard of this.
It's actually not new. It's been around since 2002. I think even earlier that, but it's called the Anaerobic Digestion Model 1. There are all different types of things that you can use to like model it, but right now I'm using Python and Visual Code. I don't really have like a link. I mean, I could show you my screen to give you an idea of like.
Let's do this. Let's, you know, run it past. This is an agenda item for next time. If you want to look at it now briefly, or can you talk about it next time maybe?
Yeah, I'm honestly open to doing all of it, so I have no restrictions. I'm, you know, so
We do a
I'm
little
down.
now and then more later. I think that's good.
Okay. Sorry, what'd you say, Margaret? A little bit now and more later?
Yeah, exactly.
Yeah.
A little crudité, hors d'oeuvre.
So I actually was doing some, it's been quite a pain, but it's been a great learning experience overall. So let me just get out of here. Let me get out of this. Basically this is an anaerobic digester. It's a continuous batch reactor for wastewater treatment plant. Right here, this digester in it is basically when we think of like when we seed the digester with microbes and the wastewater, this is what this is going to represent, this digester in it. So that's what it's called, digester initial. And then below it, this define ADM1 model. This is the model, the entire model for all of your acid-base chemistry, your fractions. The anaerobic digestion model really goes off of COD, so chemical oxygen demand, and it really starts out from carbohydrates, lipids, and proteins, and then your model will break those main factors down. So this is going to be our kinetic parameters. I can get more into that later on. Physiochemical parameters, so more of our acid-base chemistry, and then this is the size of the reactor, the gas, and pH limitations, just because, right, an anaerobic digester could only operate at certain temperatures. So for amino acids, carbohydrates, and lipids, you have to set certain pH limits, and upper and lower limit. And then you have your inhibition functions right here. You know, and then these are all your process calculations. So this is what all of the equations, monad kinetics that you start mapping out. And then right here is where you're going to put your influent. And this is where you kind of play around with what you're going to, like what exactly your substrate is going to be. I've been doing a lot of food waste and aerobic sludge. So I've just been playing around a lot with it. It's pretty intense, but it's good. I mean, yeah. So it's called Anaerobic Digestion Model 1. There's all different models, the benchmark model 2. There's a lot of research going on with it, but still a lot of work to be done.
So you can model the digester, and I suppose, you know, you can then let's see. Sorry, I got distracted there. Model a digester and then run a digester and see if it ends up doing what it said.
Yeah, there's a lot of research behind it. So you collect a lot of your data, a lot of your influent substrates. Mine will be, I'm going to be doing synthetic wastewater, but I'm going to be making synthetic wastewater that is a little more curated to like Alaska Native diets, right? So there's a lot of playing around, but once I get that data, I can break it up into this influent, and then you compare the model to the data that you have. And there's a lot of really good research that, you know, this model really does capture a lot of what a system would do, right? There will always be errors when it comes to modeling, but overall it does a really good job.
Yeah, this is awesome. I mean, it's just doing mass balance iteratively for you. That's awesome. You're very clever. I am never going to attempt to try to learn coding, so I'm impressed. But I see what you're doing there. And the first thought I have is like, we have pretty good ideas of what's going on with our synthetic poop. And then, you know, talking to the USDA folks last time, the whole idea to like get funding through this like public-private partnership is maybe like getting Trident Foods interested in like funding our study and everything. And so now it's not just like synthetic waste, it's like putting fish bits in there. And I wonder if, you know, if it wasn't too much to get like, okay, here's our synthetic poop, if we could like run it and see if like having a bunch of fish protein in there is like not going to be helpful. I mean, it'd be kind of cool to get like a proof of concept go no go thing out of a model before. I mean, obviously, whatever barrels we make are going to be slightly different, but and as a PhD student, I'm sure you have lots of extra time for stuff like this. So never mind. It's just a thought I had.
Yeah, no, that's great. I definitely will be using like synthetic, but I'll also be using, so we have connections here at the University of Kansas. My advisor is really well known within wastewater. Belinda Sturm, I'm not too sure if any of you know her, but Curtis is the one who knew her the last time, which was great. But so I will be using wastewater. Unfortunately, the only problem that I have with that, of course, is that I just don't think that it represents exactly like what it would be my aim for a rural community, of course, right? So I do have some limitations there, but I totally agree with you. That's exactly where I was going to go with it for sure. Yeah, I'm excited. There's a lot of work to be done, but I'm very motivated individual, so it's exciting.
Yeah, no, I'm stoked that you're getting to do that. And yeah, I mean that's always one of the hardest things is like trying to be like, okay, so what is a normal diet, right? Like, and that's a thing that changes seasonally, it changes community to community. So yeah, that's awesome.
Thanks.
I've got some great ideas for future agendas, and I'll circle back to people who could maybe bring some of this stuff. For example, I think it'd be really cool, Jasmine, to see some pictures and other information, maybe on the Alaska Pacific University, you said, running the Jasmine Perea commemorative digester.
Yeah, no, yeah, definitely. I have some pictures. That's hilarious. Yeah, I don't know.
Anything, pictures, performance summary, like a short spiel on it would be, I think, really helpful.
Yeah.
just like, and then you mentioned this thermokarst thing. That's an icy lake, basically, right?
Yeah, that produces methane. I'm really interested in collecting microbes. There's some work that's been done at UAF through, I believe her name is Kathy, or Katie. I might be getting that mixed up. Been trying to get in contact with her because she has a lot of work with thermokarst lakes. But yeah, I would like to collect the microbes from those, the psychrophilic methanogens, and then inoculate it into an anaerobic digester, which is exciting because I feel like I talked about that at Alaska when we were all having the conference. So yeah, that's my goal. That's my goal. If I don't have the resources to do that though, I've found a company that sells the same microorganism that she has found within a lake. So there are some loopholes that I have to figure out just because I'm located in Kansas. But yeah, that's okay. I'm sure I can figure it out. Yeah. But that's
Mike,
exactly what I would like to do.
you got anybody that can go to thermokarst lakes and get some microbes for her?
I don't necessarily have a staff that I can just assign to that. But I mean, the consortium is it's all the tribes in Alaska. We have lots of connections all over the state. If there was something specific that somebody wanted us to get, it wouldn't be too hard to ask somebody to go collect a sample.
I'm gonna do sign language. Connection.
Yeah, no, definitely, which is why I'm excited about this group. The thing though is, transporting it here. I don't really know. I don't know the depth of the microorganisms that you'd have to collect it. So these are things, and then like when you're bringing that microbe back up to the surface, right? There's different pressures. So I would definitely have to dig a little more deeper into it, but would love to keep communicating because I'm totally open to anything. Yeah.
Jasmine, I'll send an email here today or tomorrow for connecting you and one of my mentors from University of UW Seattle. She has been studying psychrophilic methanogens, and she just went to different like bogs and fens in Cascadia and collected some things, but she's been doing that since like I want to say like 2013.
Oh,
And
wow,
so
yeah.
and that's the big problem is like they grow so slowly, like but she's definitely had to overcome that with like, okay, we got to keep these alive to send them to labs and stuff. I think she's been looking at it more from a like genetics, like who's here doing this kind of thing, but she is running bench scale reactors, and so she might have some inoculum, or anyway, she's a great resource. If nothing else, she could point you in some good directions, or talk to you about overcoming those obstacles with shipping and whatnot.
Yeah, that'd be great. Yeah, thank you.
For sure.
Part of this group, Tom Curtis, who does that too,
Tom Curtis, yes.
right?
Yes.
He's the one who presented his 4-degree C digester. So he's got these critters, and he's been going to Svalbard for 20 years trying to figure this out too. So you could use this network, use the existing conduits in this network. He's busy, but if you say, hey, this is Chad and we met at the, holler at him. I'm not saying, Nate, don't bring somebody in, like some more people into this group, but we also have existing pathways for people who've been doing this. And I know we lost her early on, but I still think we want to get Robin back from UCs if we can. But anyway.
Robin, you said Robin?
At the very beginning of this whole effort, there was a woman who was at the U.S. Army Corps of Engineers Cold Climate Research Lab, which is in Vermont or New Hampshire, somewhere up there, and she was doing cold microbiome psychrophilic methanogen.
Okay.
She's kind of long gone, but Tom, I think is Tom's still connected.
That was Robin with a Y, ROBYN, wasn't she?
She was an ROBYN. Yep.
I think she might still be on on the list of members of this group?
Yeah, I think she is. She hasn't been doing anything with this for a long time, but still.
Can you remember her second name at all? Because we might have it on the other database.
I feel like I do, but I want to make sure that
Barbados, exactly.
That's the one. Yeah.
I'm in Barbados.
Yeah, similarly, Dr. Heidi Goff was invited to this and is plenty busy with other things. But yeah.
I dropped the link for that lab that you were thinking of, CCREL, Cold Climate Research Lab in New Hampshire.
Yeah. God bless you.
Well, I got to take off here shortly. Great. Thanks, everybody. A lot of info. I got some good notes. I'm going to think about agendas for next time and circle back to people. Yeah.
Thank you.
Yes.
Everybody.
Thanks. Good to see you all.
Yeah, good
Anything
to see you
else?
all.
Okay.
Appreciate
Take
it.
care.
Thanks very much.
Yeah.
Yep.
Margaret, I'll try and figure out some other of those friction points. There's somebody that builds, like he has a company building small scale, not micro, but like really small scale AD in like Renton, Washington, Seattle kind of area. So it'd be cool to talk to him and be like, hey, as like a guy running a business, what was your regulatory hurdles like? Because I'm sure there were some. So okay.
Margaret, I couldn't hear you for some reason.
I'm just saying that Nate's idea sounds like a good one to me.
Yeah. Renton, go pay him a visit.
Yeah. Not too far. All right. Hey, take
All right.
care, y'all. I mean, because I am this nerd, like this is such a great group to be a part of. And yeah, we will press bravely onward. Talk to you soon. Yeah.
Okay. Thanks, everyone. Bye.
Ciao.